intro, all the way to up to exercise 15 is what's in the lab practical. The lab practical is a week from today. Okay.
Yay!
Yay! See? We're not the only ones smiling about this. Testing opportunity is.
I just read it on your face. I'm like, I'll say yay.
At this point, you make me laugh on the whole thing. Anyways, though, on Wednesday, we will have lecture only, okay? I don't anticipate going over to the lab room at all. We'll stay here the entire time. As such, you know, if you're somebody who needs to have something to drink to stay awake, or something to snack on, feel free to bring, all right, because it'll be a lengthy lecture period. We do have a lecture quiz that day that will be at the beginning of the class, right, and that'll cover anything that, as far as we get up to, you know, everything we can complete up to this point. So 6, and then maybe an intro into 7. It'll just depend on where we stop today. And then we do have the lab quiz 8 that will be later in the lecture period. So I'll do the lecture first. We'll be taking some breaks. That means that, you know, you'll get a break before you actually take the lab quiz, especially for individuals who like to do a, you know, quick refresh right before quiz, okay? But that'll be it towards the end. If you are somebody who needs to make up a lab quiz, not lecture, lab quiz, then that'll also happen at the end of class period on Wednesday. So I'll stop early enough to allow, you know, individuals to be able to do that. Anybody who doesn't have to make up any quizzes, they leave early, and, you know, wait around for everyone else. Any questions as to this week? Okay. The lab practical on Monday, don't forget, we will be meeting in the lab room first. Okay, so you'll be headed over there on next Monday, and we will start the lab practical there.
Will the lab practical take the full time?
No.
Okay, so once the
After-
lab practical is done.
After the lab practical is done, we'll meet in here for lecture.
Okay.
Yeah.
Also, we're doing the last practical first.
First, yes, and then lecture, yes. Okay. That way you can data dump before trying to sit here and then lecture, try to listen to the lecture. All right. That being said, just as a reminder, a week, the Monday after the lab practical, so 2 Mondays from now, is the Unit 2 exam already. Okay, so that approach is complete also. All right. So we left off with the physical effects of water on growth of microorganisms and the requirements some microorganisms have for these specific environments. And basically it equates to, you know, these microorganisms that can survive. If you see any word ending in phials, that means they're loving. Okay, so this would be a organism that loves that high solute concentration. They've designed a mechanism to ensure that they can survive. You can't take these organisms out of that environment and expect them to survive. Just like the barophiles, you can't take them, you can't remove that hydrostatic pressure and expect them to survive. They won't, because they're designed already. They have mechanisms in place to be able to survive in and thrive in that environment. And so you can't just take them from that environment and put them somewhere else and expect them still to survive. All right, so we're going to go into a little bit about, talk a little bit about media used in the laboratory. So this is also in module 6 as you're reading through. And actually relates to the lab portion that we're in also. So when we look at media in the laboratory setting, there's different ways we can define them. So in the laboratory setting, we've already gone through the general, and I will again in the next slide, we've gone through the general media and the selective, enrichment, differential media. Okay, so that's all going to actually help prep you for today's quiz, right? But there's other ways we can define, categorize or group the media. One of the ways that we can is separating them between defined media, also known as synthetic media, versus complex media. Okay, and your book, I'm used to defined media. Your book calls it chemically defined media. Again, same thing. Okay, in the defined media, you know every single chemical that is present, right? All of the elements present and the exact quantity in that media. Okay, the defined media is actually something we don't use in any of our lab classes. Okay, it is very specific. This demonstrates a defined media right here, M9 broth. Every single one of these, you can determine the elements that are present, right? The only one that's listed as, say, a word would be glucose, but yes, we know the structural formula of glucose. So even though it's using a common name for that chemical, we still know all the atoms involved. And then of course we have the exact amount. So we know exactly what is present in this media. Now, on the other hand, if the other type of media would be the complex media, and complex media, we do know the amount of everything that is present in a way, but one of the components is of a mixed nature of what's actually present. Okay, the composition of one of the components, or one or more of the components, falls into a group where we don't know exactly what it is. It can vary lot to lot, such as extracts or digests of something. So, a pancreatic digest of, yeast, something where they've taken enzymes, they've broken down a particular item, and whatever's that. We know it's a mix of different amino acids that are present, but how much, we don't know. It's like taking chicken, right, and making chicken stock. Right, you take a chicken carcass, you can weigh out the same amount of chicken, put it into water, right, into exact same amount of water, boil both of those in two different pans, boil them both, and there would be slight differences. The chemical composition from one to the other is based upon lot, right? So that's where complex is recognized. So when we look at, say, tryptocase soy broth or McConkey agar, you'll notice pancreatic digestive casein, so that milk protein, enzymatic digestive soybean meal. We know how much is present of those, right? But we don't know what is actually in that, okay? And then McConkey, you see peptone, the proteose of peptone. These are all things of which we can't necessarily define. It'll vary from lot to lot, okay? And therefore it's recognized as complex. The media we use in the lab, all of them fall into complex media for our particular purposes, the basic microbiology. And then this is just a reminder again what we did in lab. So we have our general media, we have our enrichment media, selective media, and differential media. Medias can fall into more than one classification, right? So a general media is going to support the growth of a large group of organisms. And we could have a general media that is differential, assuming it has something in it that allows us to differentiate between different organisms by some sort of characteristic that the organism has, usually a metabolic characteristic, okay? Enrichment media is where we are providing, specifically adding something to the media, such as growth factors, vitamins, that we wouldn't necessarily, find in every source. And those are going to promote the growth of certain organisms, so therefore enrich for those organisms. As compared to selective media, actually is something where we add something that generally inhibits the microorganisms. So by inhibiting one group, we are then selecting for the other. okay? All right. Now, moving into then, so that has to do with growth of microorganisms in the laboratory and the medias that we use to do to provide the nutrients for them. Now, we're going to talk a little bit about the biofilm formation. Biofilms are a complex, is this complex relationship between numerous organisms, right? That is where we have a very highly structured community. The organisms can fall into different species and genera. It doesn't have to be limited just to prokaryotic cells. Oftentimes they are prokaryotic and eukaryotic living together in this biofilm. In the biofilm, the cells are both structurally and metabolically different than how they would be if they or when they are free living on their own. Okay, so as part of that highly structured community of the biofilm, they are, the cells are actually metabolically and structurally different. So they are different than what they would normally work in it by themselves. And the biofilms can form on many different surfaces, right? Or they may be free floating. So when we're looking at, say, you guys going into allied health, where the concern with biofilms would be for our medical devices, okay? And of course, if you're going towards dental care, right, plaque in the mouth is a form of biofilm that can form. Biofilms generally, and so in general, they are not beneficial for the surface that they attach to, okay? In general, they cause damage to that surface that they attach to. And therefore, there's a lot of effort and concern in making sure that they do not form. Now, how does formation occur? You have, this actually is going to go through a little, description, steps 1 through 5 of how, it establishes. But you have your first colonizers. Your first colonizers are going to attach to the surface, and that can reverse. They may, leave that attachment, or they may stay. But as they remain there, should they stay there, they're going to start producing this extracellular polymeric substance, so as this one is describing as EPS. So basically an extracellular matrix, okay? Something like a glycolax type of structure, right? Then that structure is going to end up being a large portion of the biofilm, but it's going to allow for attachment of other cells. It is, as well as it's going to set up a platform, in a way, for these group of organisms that will have, say, water channels to allow nutrients to go to get to deeper regions of that biofilm, get gases and nutrients so that we don't have necrosis. As we have cells pile on each other, eventually the ones in the center become necrotic because waste products start building up. But this extracellular matrix that gets set up is actually going to provide that a structure that allows for then these nutrients to come in, to maintain those organisms towards the center, so it doesn't become necrotic. They'll bring they'll wash away, help put removal of the waste products, and they will bring in water and gases to that environment. Now, a biofilm will also, these cells do communicate with each other through something called quorum sensing. Quorum sensing are these molecules communication molecules that allow that biofilm to regulate the number of cells that are present, looking at cell density, and so that if necessary, and the density is overwhelming for that biofilm structure, then we can see these cells depart from the biofilm. And again, once they depart from the biofilm, they become, again, free-living organisms, and therefore they'll go back to their structure and metabolism that we saw prior to joining that community. Okay? All right. Then we're going to go into, this point, as Nick was brought up, our cell division process for these prokaryotic cells. Prokaryotic cells, specifically bacteria, are going to divide through a process called binary fission. Okay, this process is a bit different than mitosis and meiosis, which is what we see with eukaryotic cell. Okay, this class we're not going into mitosis and meiosis, so if you remember it from general biology, you can say we're not doing that here. We're sticking just with binary fission. Okay. And binary fission is a little less complex than mitosis and meiosis as far as cell division is concerned, because of the fact that we don't have that membrane, right, that surrounds, that nuclear membrane that surrounds the chromosomes. So we don't have to have that membrane be disassembled. We also don't have mitotic spindles and lining up. Bacterial cells are haploid, so we're not worrying about lining up with chromosomes, matching, chromosomes, etc. before we have the cell division. All that doesn't exist here. So when we are looking at DNA looking at binary fission, the first step is going to be DNA replication. Okay. Now, at this point, we're not going into details for DNA replication. Okay, only because that's part of the module 7, so we will be moving in that direction. But for now, right, we just know that we're just going on the understanding that our circular piece of DNA that is within the cell and the nucleoid, which is just a region within the cytoplasm, is going to start that process of DNA replication. There is the understanding or belief that there are several proteins that remain attached at the origin of replication that interact with proteins that are in the cell membrane, and that is how the 2 copies are then, as replication occurs and the cell elongates, that's how the 2 copies are brought to the other side of the cell, to opposite sides of the cell. Okay, at which point then we're going to get division of that cell. Division of the cell is going to be formation of a septum. This is a little bit different than what we see in, say, a plant cell. At this point, the only other type of cell that you guys have looked at in your education in regards to a cell that has a cell wall. Okay. Do you remember from general biology, a plant cell has a cell wall also? Do you remember how it went through that cell separation? For a plant cell. Animal cells had the cleavage furrow, right? That was, we had the microtubules and a contractile ring that separates at the metaphase plate and divides a plant cell. So an animal cell, right? So you have that cleavage furrow. And then it has to do with, specifically with the cytoskeleton, a portion of the cytoskeleton creating the contractile ring that then closed off, separating, right, through cytokinesis, the 2 bodies of cytoplasm. But how did it happen when we talked about plant cells in general biology for dividing these? Because plant cells have a cell wall, right? Nobody remembers? All right, so for a plant cell, what happened was that, because we have that cell wall we need to create also. So the endoplasmic reticulum, right, created the cell wall components, and we got little vesicles, and they would start lining up at the metaphase plate. And as more and more lined up, they would fuse until we got the cell plate, right? Eventually it became a full and as more and more it continued to add on, it separated, okay? To do this, to create that cell plate, you needed the vesicles with the inside was the components for the cell wall, the cellulose structure, right, that polysaccharide present. But you need those vesicles lining up, okay, at the metaphase plate. And then fusing together to get bigger and bigger and bigger until eventually they expand the length of the cell, separating the cell. When we look at prokaryotic cells, we have a problem here. We don't have the ability to create these vesicles. We don't have membrane-bound organelles. We don't have an endoplasmic reticulum. So they work differently. They are going to have a cytoskeleton structure with a contractile ring that's going to draw in and eventually close off the cytoplasm. But as it's drawing in the cell membrane, we also see the formation of the cell wall. So it's bringing cell wall and cell membrane inward. That is referred to as the septum. Okay, and that then divides that cell, eventually closing it off completely. Okay, so it's actually a little closer to what we see in an animal cell, except the cell wall is present, and therefore the cell wall is also coming in at the same time as the cell membrane. Now, with cell division, let me stop there for a moment. Oh, well, no, I guess I could bring it up here. Here they talk about then the cell separates, right? However, this cell separation, binary fission, comes into play also with the arrangement, right? So what are some of our arrangements? We have singles, and that's demonstrated right here, right? We have doubles, and I'm gonna put chain, because really a double would just be two, chain's gonna be a longer length, right? And then we have what? Clusters. This is gonna be different, clusters. So single is if at the end of binary fission, the two cells, once the cytoplasm has been closed off and cell membrane is closed off and that, they actually separate, okay? When you talk about doubles and chains, what happens is at a dividing cell, so here's our dividing cell, closes off, it separates, but doesn't, it separates those two cytoplasm, but it doesn't actually separate and disconnect. Okay, so those two cell walls are still connected. Okay, that's a double. And if a chain would be, let's say this were to continue, right? Now all of a sudden I've got a chain formation. And in this case, so now I've got a chain going on. So there is the divide, each time division occurs, it always occurs on one axis, right? So each time the cell divides, it always divides on the same, say, y-axis. But the cells don't separate, so we get a longer and longer chain. In the case of a cluster, instead of dividing on one axis to give us then a chain formation, the first cell division might be on one axis, the second cell division might occur on a different axis, right? And so we have a series of axes going on where cell division doesn't always occur in the same direction, and therefore we get detachment doesn't happen, and therefore we get the cluster formation. So here's again where we talk about the fact that our cell wall, and in this case with binary fission, does play a role in the arrangement of that cell. Now this particular textbook got a little bit, extra information here than I normally go into with cell division as just a kind of a descriptor of. So That division, they're just demonstrating what's happening for that. It initially starts out as kind of a cleavage furrow. We have a contractile ring formation produced by a group of proteins. They create that contractile ring, bringing that cell membrane together. And as they do so, the cell wall also, we see formation of the cell wall being brought in order to make that divide. In this case, we are seeing the beginnings of maybe this would be a double, but the potential for a chain depending upon how long we continued, right? Because even though they are divided, they are still attached. Okay. Now, binary fission follows the same concept as we see with eukaryotic cells. So the prokaryotic cells go through the same sort of concept as the eukaryotic cells, and that is one cell divides to two, right? Two cells into four. So it's a logarithmic growth, exponential growth process. And there is a means by which we can determine the number of cells in a particular culture if we know some basic information. Okay? If we know the number of initial cells in the culture to begin with, and the number of generations that have occurred, if we take the number of generations to making it an exponent of 2, okay, so 2 to the exponent of the number of generations, and we multiply those numbers together, we're going to get the number of cells in any given generation. Okay, so very basically, let's come up with a problem here. Very basically, if I have a culture, and my originating culture consists of one cell, which is highly unlikely to ever happen. Usually bacterial cells are many more than one, right? So I have a food source, and one E. coli cell got onto that food source, right? And that food source sat in a nice warm environment, say under a heat lamp, for about 2 hours. Okay, so for 2 hours, we had an incubation time frame, and E. coli replicates, or goes through its generation time, right, is it divides every 30 minutes. Okay, so I already have at this point 2 of my 3 unknowns in order to solve this problem. So the number of cells, okay, at the point in time after those 2 hours, right, is going to be, well, I only had one cell to begin with, and I'm going to times that by the fact that I have 2 to the nth, right? What is my generation? That's the question. How many generations have passed after those 2 hours?
You have to divide the 2 hours by 30 minutes.
Mm-hmm. And you come up with?
4.
4, right? So you come up with 4. So I have had 4 generations over the course of those 2 hours. Because every 30 minutes, it went through cell division. Okay? And so 2 to the 4th is what?
I think
16. Yep. So this would be times 16. So at the end of the 2 hours, you'd have 16 cells. You say, that's not a big deal. No problem. Except, again, we know when we get bacteria on our food source, somebody forgot to wash their hands, they prepared the food. It's not one cell we're talking about. It's usually thousands of cells, okay? Or hundreds of thousands of cells that get on the food source, which then sits under the heat lamp, nice cozy, incubating, and dividing every 30 minutes, right? So it can get to be a very big number very quickly. Thus the reason why, say your fast food restaurants, a lot of experimentation and of course food regulation has come into play in order to ensure that anything that when they prepare food, it can only sit underneath the heat lamp for a certain period of time. Okay. Nowadays, with as much regulations that they have on your fast foods, you're less likely to get sick from your fast foods than you are if you go to, say, a salads place, right? Where you have like a salad bar, where you have the open face, right? Because most of salad is not cooked. So once contaminated, it can sit for a period of time, and at room temperature, bacteria such as E. coli is still growing. Salmonella would still be growing at that, maybe slower, but still growing, okay? All right, so when we are looking at this process, the generation of these cells we have our generation time. The generation time is the time it takes an organism to go from one cell to go through the entire process and divide into two. Okay, that is the generation time. That generation time, it does have optimum times as well as times where it's slower, right? And that will oftentimes have to do with the environmental conditions, the nutrient sources, whether the pH is right. Are we at the optimum pH, optimum temperature, right? And where our nutrient source is at. All right. Now, when we do experimentation, and we are looking at a semi-log scale, because it's easier for us then to work with, right? If I want to know how many cells I have at, say, in this case, say 11 minutes on a rhythmic scale, right? I would have to go up and up and up, but this scale is going like this. Right, it's the exponential growth means it's going up fast, not moving forward very quickly. Okay, so it could be somewhere, past by the time my line reaches this line, it could be somewhere, graphically, I'd have several sheets of paper or on up into, above the rooftop as far as where we're at, and before my line hits that exponential growth on the arithmetic scale. So instead, by plotting it on a semi-log scale, so notice we have that log scale on our y-axis. This allows us now to get a linear line, so of a constant rate increase, so that when I do look at, say, 11 minutes, I'm actually going to hit that curve and be able to determine and identify the number of cells that are present. So when we are working with this type of exponential growth, you will see us working with the log scale. That is what we're going to be looking at next when we look at the phases of the 4 phases of bacterial growth. So they set up an experiment with many different microorganisms, and what they found in the laboratory setting, and what they found was there was a very consistent graph that was showing up, okay? And it looked as follows, okay? When graphing the microorganisms at any given point in time, they do a count of that culture, right? And what they found was a graph something like this, okay? And you'll notice that they do not give a time here because the time would depend upon the organism itself. Some organisms, like E. coli and Staph aureus, have anywhere from a 20 to 30 minute generation time in optimum conditions, while other organisms in optimum conditions can take 2 weeks or longer, right? So it depends upon the organism as to what that time frame is actually going to be. But so the x-axis with the actual logging in the exact times, it's more of time increasing okay, as we go from left to right, where the start point is when the culture was introduced to the fresh media, okay? And then, of course, our number of bacterial cells in the semi-log state, using a semi-log graph, is how we're drawing our points. So, right after inoculating this particular, media with fresh organism, they took a sample, plated it for enumeration. Okay, and every so often they'd repeat that process. Let the organism grow, but take a little bit to enumerate, so that they could plot the growth of that organism over time. Okay, which is, again, for a unicellular organism, growth is not size, it's population. And what they found is this growth curve that may remain consistent no matter which organism they were looking at. Okay, so then we started looking at what was happening at these different stages of this growth curve. So the first stage of the growth curve, after just introducing the organism to fresh media, you have a period of time where the number of cells do not increase. Okay, this is referred to as the lag phase. Okay, and you might say, well, nothing's going on there, but that's not true. There's a lot going on there. The organism just was transferred from one culture tube to where they'd been for a while, okay, to a new culture tube with fresh media, right? Fresh nutrients, optimum pH for that organism. And therefore the organisms had to switch gears and move into a state where they were going to be taking in nutrients and metabolizing and breaking down these nutrients to provide the subunits for when they're going to grow. Okay, so we need nucleotides, we need amino acids. They're coming from a source, right? And that's what's happening during this lag time. We're not going to see an increase of the number of cells, but there's a lot of activity going on here as the culture switches gears, getting ready to go through cell division. And then we enter the log phase. The log phase is that where we see the highest amount of increase in population. So the culture is rapidly dividing during this period of time, using all of those subunits that they had acquired during the log phase. And theoretically, right, we would expect to see the log phase continue, except that we know in a test tube of limited resources, at some point, just due to those limited resources and metabolism, the pH levels are going to change. The access to nutrients, those fresh nutrients, are going to change. And therefore, we reach a point in the growth curve where they saw a plateau happen. The plateau, it can be referred to as the plateau phase or the stationary phase. And this is where, again, lots of things going on. It's not a slowdown of anything, but what's happening at this point is that cells are dividing, but equal number of cells are dying. Okay? And therefore, increase of population has stopped. So there's still a lot of activity going in this stationary phase. Okay? So don't misunderstand the term stationary. Stationary does not mean that we have stopped. It just means that the culture is going through at that transition. And again, it's usually due to nutrient sources and the environmental conditions having shifted, where those cell division and cell death is equal. And then we move into the death phase. The death phase is one in which the number of cells dividing is going to be lower than the number of cells that are dying off. Okay. Generally, in a test tube of media, you don't usually see a complete crash down to zero. What happens is some of these microorganisms, what they do is they're going to go into like a hybrid state, right? They're not happy and content, but they're no longer dividing, so they can't contribute to the number of increasing cells, but they're just existing. And then some cells are dying off, right? And therefore, we see that decline. But if we take a little sample from that, like a loopful from media, usually by the time you guys, the cultures that you guys are getting, when you are working in the lab, they're already in this, between this stage and this stage somewhere, okay? And so you transfer it into fresh media, and we're back at the beginning of that growth curve process. For the purposes of a commercial type of use for microorganisms, this is going to be, this stationary and death phase or decline phase is not going to be of benefit. We want to see how we can keep these organisms in the log phase. And to do that, what they have designed, of course, would be something referred to as the chemostat. The chemostat is going to be this kind of an open system. A test tube is an open system, okay? So don't get me wrong there, it's not a closed system. It's a closed system to some things though. It is a closed system to fresh nutrients. It is not a closed system for gases. So it's not a complete closed system. However, a chemostat is a complete open system because your culture is growing in this flask, and as time passes, you're continually adding fresh nutrients, fresh media to this flask, as well as there'll be a discard portion, where older media is being removed and cells are being removed. So we're keeping our population so that it doesn't get to be too much. We're removing waste products and cells. We're adding fresh nutrients and adding gases to this container. And by doing so, we can keep our culture in the log phase. Now they'll do this, as I said, in some of your commercial environments where, say, they've transformed a bacteria or a yeast to produce a product, such as insulin, okay? And they'll then take the effluent that from this chemostat, which would be considered the waste product, and they will be able to purify out the insulin, and that gets packaged up and sold and provided to individuals who need that particular protein as a medical treatment for a condition that they have. And that is one of many different means by which antibiotics, same process. Where the culture is making the antibiotic, and then they are removing the effluent, purifying the antibiotic out of that effluent, and packaging it for use. Okay. All right. Last section of this particular module is going to talk a little bit about measurement of bacterial growth. How do we do this? We can measure by doing a direct count or an indirect count. So we've already passed this stage in the lab as far as performing the experimentation, but you're still going to be next Monday, you'll still be tested on it, so this is still something that's worthwhile discussing. And it's worthwhile make sure you're reading it and that's in your textbook. It's going to provide additional support to that for that information. Okay, so a direct count is when we're directly counting cells. Okay, now we can do this through a process of using a hemocytometer, which is a special microscope slide that has a little grid pattern on it. You place your sample, a set volume, onto that microscope slide, and then you literally look under the microscope. You'll be able to focus on the grid, and you do the counts, much like you did the counts for the colony counter, only that was on using a petri dish, right? This is using a slide that has that grid pattern. And so you literally count the cells that are present. Now, in general, you're counting all cells that can be recognized, that are intact, right, that can be recognized as cells. And therefore, we are looking at viable and potentially non-viable cells. We can't say for certain whether they're viable or not, right? We know that they're there, but we don't know if they have the ability to reproduce. So we're getting a count of all cells. Another option is the Coulter counter. The Coulter counter uses electric current. So as the cells move into this little chamber here, it breaks the current, okay, and that counts. So at each time the current is broken and cells move in, it's counting it. 1, 2, 3, 4, etc. Something else that's out there that this specific book doesn't talk about would be flow cytometry. And flow cytometry is similar to the Coulter counter, except that it uses UV light instead of electrical current. Okay, but the idea is still the same. The culture passes through in this thin, narrow tube, and every time it passes through, it breaks the connection. Or in the case of the UV light disappears as the cell passes, right? And so the detector says, every time the UV light seems to have turned off because the cell got in the way between the light source, that UV light source, and the detector. Again, for this, we are counting all cells. We don't know if those particular cells can grow, so we don't know if they're viable or non-viable. Okay, we're just counting everything. If we want to do a viable cell count, then really what we need to do is make sure we let the organism grow, right? The only way that you're going to get an outcome in that case is the cell grew to something else, right, into a big mound of cells, and therefore, you know they're viable. You can only count the viable cells. You'll never count the non-viable cells because they wouldn't have grown. We did this, one of these processes in lab, that is the standard plate count. If you're doing the standard plate count, it's usually paired up with the serial dilution. And you transfer, after making the dilutions, you transfer your organism to the Petri dish. Now, we did the pour plate method of that standard plate count. That is, we put the organism in first, then poured in the liquid broth. You don't have to. You could already had prepared Petri dishes. You put it on top, and then you spread it over the surface using a specialized spreading tool. Either way, your goal is then to have separated out, found a dilution that was low enough so that you can get a count of microorganisms growing on that plate. Those microorganisms would be identified as mounds of growth, which we refer to as the colonies. Okay? And each colony represents, from their original culture, represents a single cell, or in some cases we recognize that sometimes it's a group of attached cells, where if we weren't able to completely break up the clusters into an individual cell. Another process is most probable number. Most probable number is more of an estimate. This is one in which we take our original culture and we're going to do 3 dilutions within 1/10 of each other. So 1 to 10, right? 1 to 100, 1 to 1,000. And then we are going to inoculate test tubes, 5 test tubes for each of our dilutions. In this case, they're actually going up they're saying 10 mil, 1 mil, and 0.1. Okay? And so, and you are inoculate 5 of each media, so 5 test tubes of each media. You let that incubate. After incubation, right, if the organism was able to grow, so there were cells present, they were able to grow, you're going to have some sort of differential component to be allowing you to recognize that cell growth occurred. So in this case, if we're talking about changing, the media should change from a red to a yellow color. So we would say 5 tubes are positive here, 2 tubes are positive here, and the last on that 0.1, none of them were positive. This gives us what we call a MPN index of 520, 520. And we would go to a table, a pre-established table, and we would find that index on the table, and the table would say, that combination, 520, gives you, 32 cells in the original media. So it's an estimate based upon a recognized statistical evaluation that was performed that says, when we get this combination, this is the number of cells we usually see, okay? Thus the name most probable. It's a statistical determined. Both of these mechanisms are ones in which we are looking at, again, viable cells. It's only going to show us cells that have grown and therefore were able to reproduce. One that I wish that this text, I didn't see it in this text, maybe I missed it, is also filtration. This is very common where I work, right? Because we're working with drinking water. We want to make sure that there are no more than, we want 0 total coliforms within 100 mils of drinking water, right? And that's the regulation. So in that case, if I want to process, this is going to be attached to a vacuum. Okay. If I want to process 100 mils, this is not realistic, nor is this. These are more realistic for smaller volumes. But if I want to process a large volume like 100 mils, what I'm going to do is put the that 100 mils into a funnel. Now there's a disc here that has pores, so there's a membrane filtration that has pores, okay, little holes. And I'm going to put my 100 mils, so I put my 100 mils inside this filtration unit, turn on the vacuum, and of course the water is going to go into the, catch flask. Once all the water passes through the filter, I then take this filter and I have a Petri dish where I put the filter onto that Petri dish. So the Petri dish has media on it, and I put the filter directly onto the media in the Petri dish. And I incubate it. Okay, so by doing that, I've condensed, right, in a way, instead of diluting, I've actually condensed 100 mils. Anything that was in that 100 mils is now going to be trapped on that membrane filtration. The key, of course, being that the membrane of the membrane filtration has to have pore sizes that are smaller than your standard bacteria that you're looking for. So for instance, total coliform. E. coli you've seen is about 1 micron in length, half a micron in diameter. So the pore sizes are usually 0.45 or microns or even a 0.2 micron. And therefore the E. coli will get trapped. When you set it on the media onto the membrane onto the media, you incubate it. Then what you're going to see is colony growth on that membrane should there have been any organisms present. Okay, so that's referred to as membrane filtration. And it's an option if we're trying to acquire a count of a larger volume of liquid, as compared to the other two are for smaller volumes of liquid. We're expecting a higher count number. All right. Now, the last course we also did in lab, and this is measurement of bacterial growth in an indirect count. Okay, it's indirect. The other two slides were looking at direct counts. This is looking at an indirect count. Okay, because in this case we're making a relationship connection to say high optical density or turbidity is means more cells, lower means less cells, right? And so we do use the spectrophotometer, and we determine, the count based upon a value, the optical density, and then go to a standard curve in order to identify what that value, what that optical density represents as far as the number of cells in that particular culture. This is going to detect, again, all cells, both viable and unviable. Anything that can obstruct optical density is going to be considered a cell or part of that process in making that optical density value. Including, if you're not careful and you don't set up your experiment correctly, including the color of the media itself, which is why it's very important that you calibrate the instrumentation, not with water, but with whatever sterile media, right, whatever media you use, you calibrate it with sterile media of that same in order to account for the color, so that you're removing the color from the equation, so you're not counting that as part of the cell population. All right. Since I'm going to lecture a little bit longer, I'm going to give you guys a chance. It's 7 o'clock now. I'm going to give you a chance to stand up if you want to get something to